Alright, I'd like to welcome you to this tutorial on other patterns of inheritance for Punnett squares. If you need help with any of the vocabulary, please go back and visit the model hybrid and test cross Punnett squares. It'll help you with such things as homologous, heterozygous, dominant, and recessive alleles. All these Punnett squares I'm going to talk to you about are considered monohybrids. They're just different forms. So the first thing is what's called incomplete dominance. So normally when you see a um, dominant letter, as in this case with the red flower, with a recessive allele, you would get red. But in this case, we have going on, which is called incomplete dominance, where the dominant is not dominant to the recessive. So you have a blending of sorts. So we're going to take a red snapdragon with a white snapdragon in this case, and then we'll end up getting pink flowers, which we see in the cross here. Okay. And then we also see that, so that means anytime you have a heterozygous genotype, you're going to end up getting pink flowers. All right. This mainly happens in plants, but you do see it in humans where you have sickle cell anemia. Sickle cell anemia is just where an individual, that if they're heterozygous, they would have one normal red blood cell versus a blood cell that is sickle cell, meaning it's in a different shape. Instead of being a nice round shaped, it's more in a sickle shape, like so. All right. And this is an example of incomplete dominance, but you'll perform it just like a Punnett square. So as in this case here, we have a problem here, carnations, uh, flower, the color red and white. So we take a red carnation is crossed with a white carnation. All right, so we know that they have to both be homozygous. So we know that we're going to have this crossed with this. All right, what would be the result of a cross between? So we know that that first one, dun dun, offspring, and they're all pink. So we know we get an offspring of this. Okay, and that means that's pink. So what would be the result of a cross between a pink and a white carnation? All right. So let's do this one real quick. So we have a pink. So let me just set up my Punnett square here. All right. So we have a pink, and then we have a white. All right. So we know that this is going to be pink. This will be white. This will be pink, and this will be white. So we would come out with a 2 to 2, right? So we would come out with 2 pink to 2 white carnations. What I would like you to try to do is pause the video, try to do 2 and 3, and then also try to do problem 2, which is the next slide here. Okay, so try to work out these problems before class. Let's go ahead and move on to another type of other patterns of inheritance. Another one's called codominance. So this one's a little bit different from incomplete dominance, where incomplete dominance you had a mixing. So codominance, instead, we're going to have we're going to see equal parts of the phenotype. So in this case, both alleles are equally dominant. All right. So as in like with this permanent chicken here, it has, you can either be black, white, or black and white, meaning you're not getting a mixing, you're just seeing both phenotypes, both genotypes in this case. So examples in humans would be like AB blood type in humans. So like you have a person that has A blood and B blood, but if there are individuals with A and B blood, which is just a mixing. So that individual will have an AB blood type. And then you also have what are called roan horses, which are like a bluish color. Right, so here's an example. Here's problem 1. So we can see that a roan horse, if it is homozygous dominant, then it's black. If it's a mix between black and white, which are both dominant alleles, then it would be a black and white horse, which is known as a blue roan horse. And then if we have, in this case, white is also dominant, still written in 2 uppercase W's. So let's do problem 1 together. All right. So the question is just simply asking what happens when you cross a black horse with a blue roan horse. So a black horse will be 2 capital B's, right? And a blue roan horse will be a capital B and a capital W. So we'll have two black horses in this case and two blue roan. So it would come out to a 2-2. All right. So we'll have two black to two blue roan. So what I would like to do is just pause this video and do problems 2 and 3, and then also do problem set number 2 for it looks like fish color. All right. So try those problems out, see what you get. Let's check them in class when you come. So let's move on to another. This is called multiple alleles. So most of the times alleles always come in pairs, and they always will come in pairs. But sometimes you can have what are known as multiple alleles, meaning you have more than one allele for a single gene. Okay, so a good example would be blood type. So we have A blood, B blood, and then we have O blood, which is recessive here. And I mean, those can come up with a different combinations. So you can get the AO, AB, B blood, or O blood. And these are just different combinations of that blood type. All right. So examples would be blood type, eye color, coat color, and rabbits, and such forth. Here are a couple problems. So these work just the same, except we just have multiple alleles. All right. Now you got to take in consideration that this is also a codominant. So, and the reason I know it's a codominant is just by looking at this, we have a capital A for A blood and a capital B for B blood. So there's going to be dominance here, and then the recessive is going to be the O. So what we have here, let's just do the first problem together. So a man with type AB blood is married to a woman who is also with type AB blood. Determine the probability of phenotype and genotype of their offspring. So they both have AB blood. Okay, and then so we know that this offspring is going to have A blood. This offspring will be AB, this offspring will be AB, and this offspring will be BB. Okay, so it's a 1 to 2 to 1 for the, so it's a 1 to 2 to 1 for the genotype and phenotype. Okay, so we have one A blood, one AB blood, two AB blood to one B blood. All right, so the genotype and phenotype will be exactly the same, a 1 to 2 to 1. All right, so they have a greater probability of getting a child with a 2, with AB blood. Okay, so I would like you to just pause here, take your time, work on these problems, and see what you can get. Now we're moving on to something that's a little bit different. It's still a monohybrid, but this is specifically dealing with the 23rd pair of chromosomes, okay, which is known as your sex chromosomes. All right. So this one's done a little bit differently to separate it from the rest of the chromosomes, rest of what are known as autosomes. Okay, so you use what's called like a placeholder. All right, so like in this case, we're trying to make sure that we know that we're dealing with a 6 chromosome. So when you actually go to set up a Punnett square, all right, you would have your placeholders. So in this case, we have the male up top and the female down the side, and then we just fill in. Those are supposed to be Y's. And most of the times in any Punnett squares, mass majority of times you're going to follow it on the X. There's just a lot more genes present on the X, so most of the traits that we're going to follow are X-linked traits. So this explains why certain diseases like color blindness here, why it's more prevalent in males, because they only have one X, and it is a X-linked gene. Same thing with hemophilia, same thing with Duchenne muscular dystrophy. Right? So let's see if we can try one real quick. All right. Oh, these are also more examples of X-linked traits. So these are dominant disorders. These are disorders that would be expressed equally, almost equally between male and female because they're dominant. So like rickets and Fragile X syndrome. Alright, so here we have a problem. So we cross a carrier female with a normal male, determine the genotype and phenotype. So let's just go ahead and set up a Punnett square down here. Okay. So remember, the male has an XY chromosome and female is XX. Alright, so we are just going to fill in the corresponding Punnett square. So these two would be females, this would be male. Right, and then so we're mainly looking at color blindness, and this is just followed on the X, as we can see up here. Okay, so let me get a different color. So we have a carrier female. All right, anytime you see the word carrier, that's going to mean heterozygous. So I'm just going to put a capital C with a lowercase c, and I'm going to draw a line just so that we can see the difference between a dominant and recessive. And it's with a normal male, so capital C. So if I fill this in, that's going to be a normal female, and then this will be another carrier female. This would be a normal male, and this male would express color blindness, so he would be colorblind, meaning he'll have trouble in the blue and red determination, okay, and green. If we just determine the genotype, so if we just look at female, all right, let's just look at female. So we pretty much would have a 1-to-1 genotype, but we'd have 2-to-0, pretty much just two normal females. All right, so these two would be normal. One would be a carrier, but should still be considered normal. While the males, we would have a 1-to-1, one normal to one being colorblind. Okay, and then also their genotype would be 1-to-1. All right, so please just take your time to work through the rest of the problems, see what you can do before class, and then we'll check them in class. All right, so let's go ahead and check out the last one. This one you will not have to perform a Punnett square, just because they're very complex. This is where you get into addition and multiplication rules to be able to solve these. So as you can tell, this is a tri-hybrid type Punnett square, which it's possible to do, but it's just easier, instead of working out the whole Punnett square, just to use some simple multiplication addition. So the next thing is what's called polygenic traits. Polygenic traits is where you have 2 or more genes for a single phenotype. So as in skin color or eye color. So this is a little bit different than multiple alleles, where you just have many alleles for one gene. Instead, this one you have many genes that code for one type of phenotype, like skin color or something of that nature. Okay, that's all about other patterns of inheritance. I hope this helps. If you have any questions, just bring them to class. Have a great day.